The overall theme of my PhD is fish hologenomics. But today I'm not going to talk about hologenomics in general, but what I'll talk about will have some relevance for hologenomics. So we recently conducted a study with the aim of testing whether our sampling procedure is able to capture the two main communities in the gut microbiome of fish. And this is what I will talk about today. So as most of you here at the center probably know, gut microbiomes or fish microbes are microbiomes of fish, and any other host are important for shaping the host's fitness. And as you probably also know, it's not like a single community. We can have skin microbiomes, or if you're a fish, a gill microbiome, and also the gut microbiome. And depending on the spatial scale, you have different communities, even at, for instance, the gut level. When it comes to fish and also other animals for that matter, the most common distinction in the gut is between transient and resident microbial communities. So the transient microbes are often called short-term colonizers. They live in the intestine for a shorter period of time. And they are mainly influenced by environmental factors such as diet, is one of the main things. And if you're a fish, then the salinity and temperature or level of eutrophication of the surrounding water. And they are involved in making nutrients available for the host. On the other hand, the resident microbes are to a higher degree constantly present on the mucous surface of the intestine. And therefore they're influenced by physical and the chemical environment on this mucous surface. And they're also believed to be influenced a lot by genotype, which makes this community very interesting from a hologenomic point of view. And as you might have heard before, the influence of genotype on microbiome composition is debatable how big that effect is compared to environmental factors. But maybe that's because we are looking at mostly transient microbes and not resident microbes. But yeah, that's mainly speculations from my side. But yeah, this community is involved in protection against pathogens and in maturing the immune system of the host, and also in nutrient exchange. So how do you collect a representative sample of these two communities in fish? So if you're dealing with larger fish, as the four shown here, what you can usually do is that you cut out the intestine, and then you cut it open lengthwise, and then you remove any digester in the intestine, and you can store that as a sample to characterize the transient community. And then you would wash the intestine to get rid of any leftover digester, and then you can collect a scrape or swab of the intestinal lining and use this sample to characterize the resident community. But what if you're dealing with a fish the size of a zebrafish, for instance, or juvenile stage of one of these larger fish? Then it's not really possible to collect the sample as we do for the large fish with the equipment we often have in a field setting. So there are, of course, a number of things you can do, because as you all know, for instance, zebrafish are a very common model organism. So what you can do is you can collect the whole intestine, including any digester present in the intestine. And of course, that reduces the spatial resolution, but it's easy to collect this kind of sample. Another thing you can do, which is often done, especially in captivity, is that you collect a sample after a period of fasting, because in that way you get rid of most of the transient microbes, and it's easy to collect the sample, but the composition might change. And this is especially true if you are sampling a wild fish, because as soon as you capture the fish and remove it from the wild, it won't get the same exposure to different microbes as it did before. So this is not ideal. Third thing you can do is, as soon as you capture the fish, you remove any digester in the intestine, and store that as a sample. And then you have the empty intestine that you store as another sample. And this allows you to do like a coarse separation of the two communities. But because you don't do any washing, you will expect some spillover between sample types. And this is what we wanted to test in this study, to see if this third method is capable of capturing two different communities, both in terms of diversity and of course compositional differences. And the last thing we wanted to do is to see if we could potentially identify microbial families that are likely to be resident. And to do this, we collected samples from two small species of fish. The first one is the invasive eastern mosquito fish, and the two other species are both endangered and native to the Mediterranean wetlands of Spain. And we collected samples from a total of 11 different locations. So what we did was we caught the fish in baited traps. Then we took out the intestine. And if there was any any digester present in the intestine, we squeezed that out, stored that as a sample, as a proxy for the transient community. And then we took the empty intestine and cut it into pieces, and stored that as a sample, as a proxy for the resident community. Then with the DNA extraction and amalgam sequencing. And this is what we based our analysis on. So in terms of diversity, we found that for all three species, the intestinal wall samples had a significantly lower diversity compared to digester samples. And this was independent on the respective level of diversity within each species. For instance, you see that the one to the right, Valencia, has a lower overall diversity compared to other species. And every time there's a green line between a sample pair, it indicates that the intestinal wall has a lower diversity compared to the digester. Then we looked at the compositional differences, and this is what you see here in some MDS plots, where different colors indicate different locations and different shapes indicates different sample types. And each pair of samples are connected by a line. Even though it might not be super clear from these plots, we found that sample type had a significant influence on the community composition, and we're able to explain 1 to 12% of the variance depending on the species. We also found that location had a significant influence on the composition and explained a much higher proportion of the variance, which was also what we expected. And as I mentioned before, the transient community is believed to be mainly influenced by environmental factors, and to a higher degree than the resident community. So we also wanted to see if this was the case for our data set. So we separated our data into sample types, depending on sample type, and then we had a look at how location influenced each sample type. And sure enough, we found that the amount of variance explained by location were higher for the samples from the digester, so this transient community, which makes sense and is similar to what has been found in other studies as well. Final thing we wanted to do is to test if we could identify microbial families that were enriched in intestinal wall samples and thereby potentially resident microbes. And the way we did this was by using joint distribution species modeling based on the abundance of these microbial families. And we found 7 different families, and these are the ones in red and orange, that were significantly enriched in the intestinal wall samples. And all the blue colors indicates microbes that are significantly enriched in digester samples. And as you might notice, some of these families that are enriched in the intestinal samples from some species of fish are significantly enriched in the digester sample from another species. And this could be due to the fact that these microbes are present in the environment but are not able to colonize all these species of fish, or an artifact of our sampling procedure. That's hard to say. So using amalgam sequencing, this might be the best we can do to try to identify specific microbial families. But in order to verify if these microbial families are actually a part of the resident community, we need to use more detailed methods. And these include, for instance, laser capture microdissection in combination with the fluorescence in situ hybridization, as you see here on the left for the mouse column. And this will tell us something about the spatial orientation of these microbes in relation to the host tissue. We'd also use shotgun metagenomics to have a look at, for instance, the genome size of these bacteria, but also like specific genes that could indicate a symbiotic lifestyle with a host. Yeah. So just to quickly sum up, our sampling method were able to capture these two distinct communities, even though it was a very simple method we used. And we found that the resident community was less diverse compared to the transient community, and the composition between these sample types. And also we could identify microbes that were potentially resident, but we need more detailed methods to verify these results. So it is, of course, valid to look at the microbiome of these small fish as a single entity, including digester and intestine and everything. But I would argue that if we want to study co-evolution between host and microbes, or if we want to develop novel host-specific probiotic strains, then we need to focus more on those microbes that are developed to survive and persist in the intestinal environment. And even though this slide could suggest otherwise, the transient microbes are not also important, for sure. But I just think that we need to include in our discussions and our conclusions that these two communities exist, because that's often something you will see that authors does not include. Yeah. And with that, I would like to say thanks to all the co-authors of this paper that will hopefully soon be published, and all the staff in the Conservation Center in Valencia, where we did our field work. Yeah, thank you for listening.
Thanks, Lasse. It's very nice to hear that. If people have got questions, please put an X in the chat. I just want to point out one thing, and I might have done this before. You are aware that one of those pictures you had was a turtle, right? Not a fish. Just be sure we're on the same page.
That explains a lot.
Yeah, so a question for you. So for every fish species, you have multiple locations, right?
Mm-hmm.
If you look at one fish species and look at all the locations, is the resident community pretty consistent by location? I know the transient one will vary, but would you also see the resident community also varying by location?
Yeah, it does.
Does it vary by
the case? Yeah.
Does it vary by as much as the transient one does, or is it more stable?
To a lesser degree, yeah.
Okay, and then, but flipping it the other way, when you have for one fish at many locations, the transient community, do you find consistently, however, certain microbes appearing in the transient one, no matter where they live? Because presumably there must be functionally relevant ones. It's like selecting from the environment, right?
Yeah, so that the modeling approach we used, it does account for differences between locations. So these microbes that we found was like, that's across locations.
Okay, so there are transient ones that are conserved by location, right? Which means that they must be relevant, right? Because if they're continually appearing.
Yeah.
Okay. Yeah, so yeah, that's very interesting. That continuity, I think, is obviously key in all this, right? For the things that are really important.
Yeah, and this approach is just meant to give us an idea about which microbes to look for.
Yeah. Awesome, thanks. Morten Limborg.
Hi. Cool stuff, Lasse. I think it's really nice to see all these thorough evaluations of the different parameters explaining variation, although it might seem very logical. It's rarely reported at this detail level in papers. I was wondering, are these species easy to keep in the lab? Because we or Edicure are kind of studying some similar questions with a different approach of like using CRISPR to knock out genes. But for us, it's basically impossible to have cool data from the wild because of they would need to go to Bangladesh or India and
So
catch zebrafish.
if the fish species is possible to have them in captivity.
It's easy.
Yeah. I mean, I think that the issue would be that like one of them is super invasive, and there might be strict rules about bringing them to Denmark, for instance, and the two others are highly endangered. See, I think Anton can add more to this.
I was just gonna say that, Lasse, don't you have some gambusias in the tank just behind you?
No, that's the regular zebrafish. I wish.
Okay.
Thank you. So if anyone else got a question, put an X in. Just a last question for you, Lasse. I know that you haven't talked about this here, but back to this question that you originally were looking at with regards to adaptive success. Do you actually see any pattern that explains the invasive mosquito fish versus the others at this point?
I mean, if we just look at amplicon data, and if we like what we could expect was that the invasive species would show a higher level of plasticity between different environments, but that does not appear to be the case. But it could also be the other way around, that the microbiome is more conserved in the invasive species.
Yeah.
But with this amplicon data, we haven't found a pattern yet. But we're also looking at some shotgun data, so maybe in the functionalities will be different.
Cool. All right, thanks.